All right, so this is a USMLE Step 1 NBME style Q&A discussion. So here's some instruction. Please click on the link in the description. You'll find this link in the Facebook description. At the same time, those who are joining through webinar, when you are done with the session, you will have that link or you'll have that web page popped up in your window. So you won't have to worry, it's an automatic thing. You just have to fill this form just for once, all right? Let's say you are joining the session every day. You don't need to fill out the form every day. You just need to fill out this form just once, right? So here's some instruction. A question will be shown. All right, let me take my pen. All right, so a question will be shown, and these questions are NBME style questions, and don't worry, these are not from the online NBMEs that you have, that you use for your step 1 preparation gauging. That means before going to the step 1, we usually take the step 1 or step 2, we usually take the NBMEs, right? So this NBMEs, these questions, they're not from the new NBMEs. Those are available online. So if you see this question and you know the answer, it won't hamper your result, or it will not cause any bias in your result. So these are from the old NBMEs, and these questions are modified, all right? So you will have 90 seconds to answer the question. So I'll show you the question. I'll give you 90 seconds. Answer it. Those who are joining through the webinar, please use your chat box here to answer the question. By the way, in your chat box, you will see that option. There is option like who you are sending this answer to. So you should choose all panelists here. All right, there's option of all panelists and attendees. Don't choose it, choose the all panelists here. So those who are joining through the webinar, you will not be biased by looking at others' answer. And in the Facebook, you can just type in your answer in the comment section, or you cannot, or you just can think by yourself, whatever you choose, right? And those who are joining through Facebook, please share this live video, all right? Okay, so that's it. So my plan is to go over these topics. Like, let's say today I will give you 2 questions from the biochemistry and immunology, all right? So this 2 topic, these 2 subjects, I choose 2 questions, all right? 2 questions I chose from these 2 topics. And these are very a primary stage topic. Let's say you are studying for USMLE, you have started for studying for USMLE. So you are not into the very detail yet. So anyone who is very beginner, who is in the middle stage, who is in very advanced stage, anyone can be benefited from this session. All right. So please share this session so that other people can be benefited. Okay, so here goes your first question, and here is it. Look at it, try to answer, try to read, try to answer, and then I'll discuss about it. All right? All right, so I got some answers, like in the chat box. I got some answers. At the same time, is there anyone in the Facebook who wants to share their answer? Is there really anyone who wants to share the answer? Like sometimes it happens that when we see study materials in the Facebook, well, we think that, oh my God, what this study material is doing in the Facebook. I'm not here to study in the Facebook, so we don't like to participate in those materials. All right, so those who are joining through the Facebook, my suggestion will be to join in the webinar if you directly want to participate, like as a real class, right? In the Facebook, it's kind of like in the social stream, you see these videos and you're like, oh my God, I don't want to see it. I want to see something else. So that's why sometimes we skip it. All right, so, but is there anyone in Facebook who wants to answer? All right. Okay, cool. So I'm going to the discussion that how to answer this question. All right. So this question is from the basic cytology, right? The cell, the molecular unit of our body. So the cell structure, if you know the cellular structure, you can answer this question. This is from the very basic of the cellular structure and cellular organelle. All right. So one thing I suggest to my students all the time that how you can tackle a question. All right. So first thing you need to do when you are thrown a question, right? In that day when you are taking the step one, what will happen? You will sit in front of the computer, and that particular computer, will throw 280 question to your mouth. I mean, not to your mouth. I mean, it's like uh, it's in the computer, and you will see those 280 questions. That particular computer will choose those 280 questions out of all the Q banks, all the questions this USMLE have, right? So out of those 280 questions, you you will have to answer all the 280 questions in 8 hours, right? Not 8 hours, 7 hours, because in step one there is 7 blocks, and in step two CK there is 8 blocks. So the thing is, when a question is in front of you, what do you do first? So first thing I used to do, and that was wrong, that I used to start from the beginning. That I used to start from, okay, here's a question. A 19-month-old boy is brought to the physician by his mother because of 1.5-week history, like this. But this is actually a wrong way to tackle a question, because these questions, they will twist and turn inside the vignette. That means you will not have a proper way, or you will not be in a proper track if you're reading the question from the beginning. It is better that you start the question from the last line, because if you read the last line, you will know that what type of question it is. What is ultimately going to be asked? Is it some pathophysiology, or is it some cause? Is it some diagnosis? Is it some treatment? You will know from the last line. If you start from the first line, most likely you will be lost in thinking, because these questions, these USMLE questions, they make it confusing. I believe that always, I believe that my students, or anyone who are preparing for step one, they are all very efficient doctors, right? You all know how to tackle these different situations in real life. Probably your knowledge is also more or less the same. But the thing is, in this USMLE, there is discrepancies in scores, right? Some score very high, some scores very low. Why is that? Because of the thinking ability. The knowledge base you have is almost same, but on that particular moment, how you are thinking or how you are approaching a question, that matters. So that's why you need to be careful. You need to know what is exactly being asked and how you can proceed with that answer. All right. So here you can see that the last line, if I read the last line of this question is, which of the following types of modification is required prior to destruction of misfolded proteins in these patients' proteasome? So by just reading the last line, I think many of you can answer the question without even going through the whole vignette. I don't recommend it. I mean, without going to the vignette, I don't recommend this thing. Always read the last line first, then go over the vignette. Be sure that you are right, and then choose the answer. So they are talking about something called proteasome, right? A cellular organelle that is responsible for kind of, you can say, trash handling, right? Inside our cell, there's the organelle called proteasome that helps the cellular trash or cellular unwanted protein to be degraded, right? It's a barrel-shaped structure, and this barrel-shaped structure is there. It's kind of like the, what do you say, the garbage truck. It has this own processing unit, and it can degrade the unwanted proteins or materials inside the cell, right? So let's go over the question now and see that what exactly it is asking for. So a 19-month-old boy is brought to the physician by his mother because of 1.5-week history of green nasal discharge and cough. All right. So it seems like that there is some infection going on, right, in this kid. He has a past history of frequent respiratory tract illness and pneumonia. So when you see the word frequent, you suspect some real grave pathology is behind that, right? And he has a 6-month history of large foul-smelling stools and a salty taste when she kisses him. By reading up to this, I think many of you can understand what this disease is. Can anyone answer what type of disease probably they're talking about? Anyone? Yes, I got an answer. Cystic fibrosis, right? By reading up to this, you can, from this description, you already know that they are talking about cystic fibrosis, because a frequent history of respiratory tract pathology at the same time, very pathognomonic findings, salty taste when she kisses him. That means what? This kid's sweat. The sweat has more salt content. That's what happens in cystic fibrosis. In cystic fibrosis, there is problem in some cellular transporter, right? In some cellular transporter, or you can say not transporter, in cellular channel, there's a problem that leads to discrepancies of electrolytes. What happens here that the salt, the sodium chloride, that's the salt, right? The salt content of the sweat, it gets increased, all right? At the same time, these patients in cystic fibrosis, this is very common in the US. That's why they like to ask questions about cystic fibrosis all the time. So you might see this question in your step 1, probably step 2 CK, probably step 3, all right? Now, this thing, the salty taste, is a pathognomonic finding because here in cystic fibrosis, we get more salty content in the, or salt content in the sweat, all right? Then it says there is no family history of similar condition. It can happen. It can be a sporadic condition. That means no family history, just a person can get it. He is at 5th percentile for height and below the 3rd percentile for weight. What does it mean? It means this pathology of this kid is preventing his growth, all right? There is growth impairment, and that is also a finding of cystic fibrosis, all right? And his temperature is 38.4 and abdominal high temperature, right? Abdominal examination shows distension because in cystic fibrosis, what happens, it is a pathology of multiple site. There is salt in the sweat, you know it already. In the respiratory tract, the secretion of the respiratory tract, it becomes thick, all right? In the gut, the secretion of the gut, it becomes thick. So what happens when through some luminal space like this respiratory tract or the gut, the fluid cannot move freely? There can be accumulation of things, materials. The fluid in the mucous membrane of the respiratory tract, they cannot move freely, and bacteria grow there. There can be infection. In the gut, the fluids cannot move freely because this gut secretion becomes thick. So what happens? The material, the stool, they cannot flow freely. They build up, they pile up, and there can be this distension and kind of obstruction in the gut, functional obstruction in the gut that leads to this distension of the belly, right? Genetic testing shows a mutation in the phenylalanine, right? PHE, phenylalanine residue, and at position 508 in the cystic fibrosis transmembrane conductance regulator gene. So this thing, this gene, the cystic fibrosis conductance regulator gene, there is a mutation that happens here. If you don't know about the mutation, that's fine. These questions, they should throw you this difficult and critical words to confuse you. But if you are done with the biochemistry, if you read the biochemistry already, you know that this mutation, F508 mutation, we call it, this is the most common mutation in the cystic fibrosis. What does it mean? It means in the genetic sequence of this gene, with the name of the gene is CFTR gene, by the way, that CFTR gene has a position that is 508. On that position, there is some mutation, and that leads to this pathologic what is a manifestation of the cystic fibrosis. And that's, you need to remember, this mutation is the most common mutation in all the cystic fibrosis patients. By the way, I use the word most common. It means the cystic fibrosis can be a manifestation of many other mutations. So as we are not discussing the cystic fibrosis today, probably some other day we'll discuss the cystic fibrosis in detail. So let's go over our question now. It says they did the genetic testing and they found this mutation in cystic fibrosis transmembrane regulator gene. And this thing gives you the idea that this patient has cystic fibrosis. All right. Now the last line, which we read already in the beginning of the question reading, it says, which of the following types of modification is required prior to destruction of misfolded proteins in the patient's proteasome? So they are describing this disease, or they are describing this vignette about the cystic fibrosis all through, right? In the last line, they're asking you about a basic cytology question. So what is that? This is the typical USMLE style confusing question. So probably if you have started from the beginning, you will think about, okay, they might ask me about cystic fibrosis. I know everything about cystic fibrosis. Cystic fibrosis causes this in the nasal tract, causes this in the respiratory tract, causes that in the GI tract. I know everything. I know even how you can treat the cystic fibrosis. You might be thinking about this if you started this question from the beginning. But as you have started the question by reading the last line, you know what they're going to ask. And they're actually asking about a basic cellular structural physiological question, right? So what they're asking the function of proteasome. So what is proteasome? Again, so proteasome is a barrel-shaped structure. Let's say I draw a barrel, all right? So this is a barrel, and this proteasome is a barrel-shaped structure. And what happens, the proteins, those are misfolded. Let's say this is a misfolded protein. This misfolded protein pass through it, and they come out as degraded material. All right, that's the basic function of the proteasome. But the thing is, how this proteasome know which protein to be degraded. It's like, you know, that when the garbage truck comes, like in front of your house to take the garbage, they pick the cans, right? And there is separate cans there, the garbage trash can. One is for the recycling, one is for just the regular trash, one can be biological household products and things, one can be for the leaves that fall during the fall, right? So these cans, they are different in colors, right? And the garbage man or the garbage truck knows that which can to be disposed in which truck, something like that. This proteasome also needs to know which protein to be degraded. It is not going to degrade everything. It is only going to degrade the misfolded protein. That means the protein that is unwanted. That is unwanted. So what happens here, this misfolded protein, it gets tagged. It gets tagged by some small molecules. Do you know what this molecule is? This molecule is called ubiquitin. These misfolded proteins, they get tagged by this small molecule. This is a signal. These tagged misfolded proteins, these molecules, these small red molecules that I drew, these are ubiquitin. When a protein, when a misfolded protein is tagged by this ubiquitin, then our proteasome guy, it knows, all right, I need to degrade that protein because it has the signal. It has this, you can say, it has this instruction for me. It needs to be degraded. So this happens before proteasome can degrade it. And that's what actually your question is asking. Which of the following types of modification is required prior to destruction of misfolded proteins in this patient's proteasome? That means to destruct a misfolded protein, the protein needs to be tagged with ubiquitin. So in your option, you have this E that is called ubiquitination. So ubiquitination happens before a protein is degraded by the proteasome. And that's what your question is asking. So in this question, what you have learned? You learned that the question can be confusing. They can be really derailing you from the proper track if you have started the question from the beginning, reading the question from the beginning, right? So my suggestion would be towards all of you to start reading the question from the very last line. It will not cause the distraction that the question might cause if you have started it from the beginning. So by reading from the last line, you know that where your question is ultimately taking you. So read it from the last line and then go into the vignette, proper vignette, try to read it and try to understand it properly. By the way, once, read your question once. It is said that if you have to read the question 2 times, it means you haven't paid attention the first time. Because when you are taking the step 1, it's stressful. I'm not denying it's stressful. This is a 7 hours question exam. 1 hour's break, and that is nothing to those 7 hours, right? So you're sitting in front of this computer and this computer is throwing you a question, and these questions, you need to think. That's the key. You need to think in every question. Even if you know something very good, still you need to think. That's the beauty, or that's the, you can say, misfortune of USMLE Step 1. That every question needs you to think. You need to think every question before you can answer. So when you are taking that test, 7 hours, Sometimes it happens you can be distressed, you can be stressed, and you might lose your thinking capabilities sometimes. So what happens in that time, um, try to go slow and try to read the question once. Sometimes it happens you read the question, you don't know what you read, you read it again, and there is some processing difficulties in your brain. So it happens that you might end up choosing a wrong answer, right? So go slow, a little slow, and not too much slow because time is a fact also here. So go slow, read the question properly, and then answer it. My suggestion would be to read the last line first and then go over the full question. All right. Okay, so now let's go to the next question. And again, same thing. I will show you the question. You will think about it, read the last line first, then come to the first, the full vignette, and then I will discuss about the topic. All right. And this is also a basic, from the basics, like it's an immunology question related to whatever we have read now. So if you know, if you understand what we have read now, you can probably answer the next question too. So this is kind of a clue you will not get in your real deal, but as this is a discussion or explanation class, I think I can share this with you. All right, here is your next question. Read it and answer in the chat box. All right, so probably I got some answer from the Facebook also, and also in the chat box of the webinar. Sorry, let's discuss. So this thing is a question again from the basic of what is that? Cytology and also the immunology, all right? Both of it is here in this discussion. So let's see what it says. Read the last line first. It says that which of the following is most likely to occur regarding the class 1 MHC products? Okay, so the class 1 MHC products, right? So this thing is it was a mystery for me. Like while I was in med school, I was like, oh my God, what is class 1 MHC? What is class 2 MHC? How I'm gonna read this and how they're interacting with the immune cell. This was a mystery for me. Then eventually I knew that these proteins, this class 1 MHC or class 2 MHC, these are what? These are some proteins that are required for a cell to process and to interact, to process and to, by the way, what you can say, to express antigen in the surface of the cell. To express antigen in the surface of the cell, these proteins are necessary. Major histocompatibility complex, simply we call it MHC. So there are two types of MHC. One is MHC1 and the other one is obviously MHC class 2, and they interact with certain T lymphocyte. So do you know which T lymphocyte the MHC1 interact with? Anyone from you know that which T lymphocyte our MHC1 interacts with? Is it a CD4 positive T lymphocyte or a CD8 positive T lymphocyte? What you say? It's a CD8 positive T lymphocyte. Very good. I got this answer from the webinars. So this MHC, they have certain ways of interaction, certain cells to be interacting with. Like MHC1 interacts with CD8 positive T lymphocyte. MHC2 interacts with CD4 positive T lymphocyte. The thing is, this MHC, their way of antigen expression is different. The MHC1 is different than MHC2. So to know this, you need to know that what these T cells are doing here. So basically, the T lymphocytes, if I write the name, T lymphocytes, they are many types, but here we are discussing two of them. One of them is CD4 positive T lymphocyte, another is the CD8 positive T lymphocyte. By the way, they have these different names. The CD4 positive T lymphocyte, we call them helper T cell, right? And the CD8 positive T lymphocyte, we call them cytotoxic T cell, all right? So the thing is, now what they're doing, so when you read the immunology, you know it better that what these T lymphocytes are doing. In a very short discussion, this CD4 positive or helper T lymphocytes, they want to interact with antigens. Those are expressed by MHC class 2 molecule. That means, let's say here's a cell. Let's say here's a cell. Probably this is your macrophage or a neutrophil, all right? It engulfs one antigen. Right, antigen means something that is that can be usually antigens are foreign, right? That is from outside of our body. We don't want it. So our macrophage, they engulf it and they will process and express it on their surface with the help of this MHC molecule. All right, this MHC molecule is MHC class 2. This MHC molecule is MHC class 2, and here on top of this MHC class 2, there will be a fragment of this thing, this antigen. And then our CD4 positive T helper cell will come and it will interact with this antigen that is presented by the MHC class 2. That's very simple. Another thing is, let's say here's a cell which is a tumor cell. Or here's a cell which is a virally infected cell. Some viral proteins are being produced there, all right? So what happens, this cell, which is already a tumor cell or a virus infected cell, this cell needs to be destroyed. And this destruction of this cell is done by our cytotoxic T cell, right? This cytotoxic T cell will come here, it will sit here, and it will try to destroy this virus infected cell. But how this T cell or how this cytotoxic T cell will know that this cell is virus infected cell, right? So it will know by looking at the MHC1 protein that this cell will express, this viral infected cell will express, along with the antigen it will express. That means this viral infected cell will express a MHC class 1 protein. Let's say this is MHC class 1 protein. I'm out of space. So, and it will present this antigen. It is present this protein that is getting produced inside this cell. And looking at that protein along with the MHC class 1, this CD8 positive T cell will do its job. It will cause toxic effect on this virally infected cell, and it will try to destroy the cell. That's what the normal physiology happens, right? Now the thing is, this loading of this antigen into your MHC1 or MHC2 is different. The MHC1 loads antigen differently than MHC2. If I give you some idea about how this antigen loading happens, you need to know the very basic of our cellular or molecular dogma of life. That means the genetics, right? So these MHC proteins that we're discussing, they are proteins, right? And probably most of you know that how the protein synthesis happen. Protein synthesis happens by passing or surpassing several steps. First, the gene. The gene needs to be expressed. That means in our nucleus, there is chromosome. The chromosome is composed of genes, DNAs, right? They needs to be expressed. From the DNA, we'll get RNA, most likely mRNA, messenger RNA, and the messenger RNA eventually will translate into protein, right? And this protein synthesis occurs with the help of something we know, all of you know, that ribosome. Ribosome takes that messenger RNA and creates the protein. MHC is nothing exceptional. It's a protein, so it will be created the way all other proteins are getting created. First, there will be this gene expression, transcription of the DNA to RNA. The RNA will eventually translate into the protein, the MHC protein. What happens in case of MHC1, that we know that here is the MHC1, the virally infected cell, right? So let's say this is a virally infected cell, and in this cell, here is the nucleus, and inside the nucleus, you know that there is this DNA material, and probably from this DNA material, you eventually get the mRNA, and that mRNA, let's say this is the mRNA, this is eventually getting translated into MHC class 1, right? So what happens, this virally infected cell, inside this virally infected cell, there is some viral protein also, right? So these are the antigens. This is not a regular cellular protein, right? This is an antigen. This is a viral protein. This cell doesn't want it. So what this cell does, it plans, all right, let's express this antigen outside with the help of the MHC class 1 so that the CD8 positive T cell comes and it can destroy this cell. And the cell is saved, right? So what happens here? This virally produced protein, they get expressed by the MHC. Whoa. But how they load into the MHC, that you need to know. This mRNA is getting translated into MHC protein with the help of the ribosome. And let's say here is one portion of your ribosome, here is another portion of your ribosome, and they are sitting here just adjacent to a structure that is called rough endoplasmic reticulum. This mRNA will convert into a protein, MHC1 protein, and this protein will eventually go inside this cell. All right. Now this is your MHC class 1 protein that is already inside your rough endoplasmic reticulum. So what happens here? Again, to recap, the gene, it converts the DNA, the gene, it converts into mRNA. The mRNA converts into the protein MHC class 1, and now it's inside the rough endoplasmic reticulum. At the same time, what happens, this viral protein, it also enters this endoplasmic reticulum through some specific transporter. And what it does, it comes here and it sits on top of this MHC's head. So that's how MHC1 is loaded with the viral protein. All right. Now what happens, this MHC class 1 with its viral protein will go through another cellular organelle that is called the Golgi complex. All right, let's just say this is the Golgi complex. And from the Golgi complex, this whole thing will be sent to the cell surface where they will express this MHC1 along with this viral protein. And then the CD8 positive cell will come and it will see that, okay, there is this thing going on. There is this viral protein expressed on a cell. Let's do my job. Let's kill the cell. And that's how the process works. All right. So as this question is about the MHC class 1, I just discussed the MHC class 1. If you can go to my I'm not sure if it is on my YouTube channel. I will discuss the MHC class 2 some other day. All right, let's just focus on the question first, then we will proceed further. So now what happens? I just discussed this because I found the MHC class 1 here. So probably you will have the idea how the MHC class 1 works. Okay, so now the question, the real thing. So it says that during an experiment, the US12 gene, we don't know what is US12 gene. You don't have to know. These are the confusing, complex terms the USMLE like to use. Trust me, all right? Don't trust USMLE ever. So here, during an experiment, the US12 gene product of CMV. Do you know what is CMV? This is cytomegalovirus, right? The CMV is expressed constitutively after stable transfection in an experimental tumor cell lineage, right? So they're saying that this virus protein, this virus, the CMV, has this gene US12, whatever the gene is, this is getting expressed. This gene product of this virus, CMV, is getting expressed when this virus is transfected. That means they put the virus into some experimental cell, experimental tumor cell. Right? So this virus is inside this experimental tumor cell, and inside the cell, this virus is expressing this gene that is a US12 gene. That's fine. All right. So what they say next? It is found that this gene product causes translocation of nascent class 1 MHC molecules from the endoplasmic reticulum into the cytosol. Well, now it's getting really intense, right? So what happens here? You need to know now this picture, what I discussed here. This MHC protein is getting translated by the ribosome and it is inside the endoplasmic reticulum. If I draw a more clear picture, then probably let's take another color here. Let's take the ink, and here you can see that, let's say this is the endoplasmic reticulum, all right? A big endoplasmic reticulum, bigger than life, right? So inside this endoplasmic reticulum, here is your MHC1 molecule, okay? And usually what happens, the virus protein, they also enter this endoplasmic reticulum and they sit here. That's what the normal physiology is, right? But in your question, it is saying this CMV thing is producing some protein that is displacing this endoplasmic reticulum into the cytosol. So what I discussed earlier, if you remember, this this MHC molecule will go into the Golgi bodies, right? And from the Golgi bodies, they will be sent to the cell surface. But what happens here, that this this MHC molecule, MHC class 1 molecule, in your question, according to your question, this thing is displaced into the cytosol. Now, they are not going into the Golgi bodies. They're not going into the cell surface. That's what they're saying. They're saying it indirectly. They could have said that, okay, the MHC class 1 is not going to the cell surface. No, they are not saying it. They are saying it is displaced from the rough endoplasmic reticulum. And you need to think, all right, what happens? If it is displaced into the cytosol rather than going to the Golgi bodies, it is not going to the cell surface. So this protein is of no use. It becomes unnecessary. This MHC1 protein, if it is not going to the cell surface, it becomes unnecessary. So what happens here? Let's see what they are asking now. Which of the following is most likely to occur regarding this class 1 MHC product? So what do you think? This MHC protein, they were supposed to be going to the cell surface from the rough endoplasmic reticulum through the Golgi bodies to the cell surface, but now they are displaced off from the rough endoplasmic reticulum. Now, what do you think will happen to this protein? I got some answer. Some of you said association with invariant chain. A is the answer. No. Invariant chain, we haven't discussed it. This is a part of MHC class 2 molecule. When you read the immunology, the basic of immunology, you know that this MHC class 2 has this invariant chain. So this has nothing to do with the MHC class 1. Number 2, option 2, binding of peptides from the endocytic pathway. What is endocytic pathway? What do you think? This thing is, okay, so I got a question. Could you provide me the link of video so that I can watch later, as I missed the initial part? Yeah, one thing is you can watch it on my Facebook page. The live will be there. And also I will upload this recording on my YouTube channel and we'll share it on my Facebook page. Do you have my Facebook page link, everyone? Those who are joining in the Facebook, you already have it. But those you who are joining in the webinar, let's just type in the link in the chat box so you know it, right? Yes, I sent the link to everyone. Did you get the link? Just click on that link, USMLE 121, and you will get the video link shared in my Facebook page. Also, I'll share the YouTube video link. So anywhere you can watch. The live will be there in the Facebook, and you can also find other live sessions that I did like long time back. I used to do these sessions long time ago, and you can find those videos also, and also this video will be there. All right. So now you get it, right? You can check in the Zoom, the webinar chat box. I shared the link of my Facebook page, so you can get it there. All right, cool. Okay, now let's come to the question. So number B is binding of peptides from the endocytic pathway. What does it mean? Endocytic pathway. It reminds you of something that some cell is engulfing something. Some product is endocytosed. That means anything outside of the cell is getting inside of the cell. That process is called endocytosis. Do you think MHC1 has anything to do with endocytosis? No, because MHC class 1 deals with MHC class 1 deals with proteins, those are produced inside the cell. MHC class 1 again, MHC class 1 deals with the proteins, the viral proteins or the tumor proteins, those are produced inside the cell. There's nothing to do anything outside from the cell endocytosis or anything. Endocytosis is relevant with MHC class 2. So as I haven't discussed MHC class 2, so I'm not going into that discussion, but just for now, you know that endocytosis is not, there is nothing endocytosis is related with MHC class 1. It is related with MHC class 2. Number C, degradation by the proteasome. Anyone want to pick this answer? Yes, I can see some of you have picked this answer for the question that what happens to that misfortunate or unfortunate MHC class 1 protein? It gets degraded by the proteasome. Yes, that's the answer. What happens that this MHC class 1 molecule, if it is not going to the gallbladder, sorry, not gallbladder, GB is Golgi bodies. If it is not going to the Golgi bodies, and if it is not going to the Golgi bodies, it is not going to the cell surface, what happens? Here, what happens? It is getting dumped into the cytosol, right? So when it is in the cytosol, this is unwanted. This is unnecessary. The cell doesn't want it. You are done. You needed to be in the cell surface. You are not there. You are in the cytosol. What the hell you are doing? Sorry for the word. What you are doing here in the cytosol, you are not needed here. So what happens? The proteasome kicks in. Remember the proteasome from the previous question? Just we discussed the proteasome, right? It degrades the protein. Those are not anymore needed inside the cell. This MHC1 molecule is not needed anymore inside the cell. Let's just degrade it. And you know what? Proteasome is here to do that job. And proteasome will do it very, very sincerely. It will destroy this MHC1 molecule inside the cell, degrade it. Whatever the last two options, just read it because you already found the answer. You are the king of the world. What happens to the next two? Formation of class 1 MHC and class 2 hybrid molecules. Oh no, we don't know that. Even if something happens like this is beyond our way of discussion or scope of our discussion for step 1. That MHC class 1 and class 2, they are combining to produce some hybrid. We don't know it. Just skip. Number E, interaction with the T lymphocyte receptors. I don't think so. If this MHC molecule is not going to the Golgi bodies, it is never going to the cell surface, and if it is not going to the cell surface, it is not interacting with the T lymphocyte receptors. So your answer is proteasome, ubiquitin proteasome degradation. Yes, in the end, does it all go to lysosome for degradation? Most likely. You're right that most likely when there is degraded products inside the cell, these degraded products need to be dissolved. At the end of everything, most likely lysosome will kick in and these degraded small products will be vanished from the cell. Or probably they will stay in the cell as small, literally non-significant molecules of protein that will not cause any problem with the cell. But probably you are right, yes. Lysosomes will come in and degrade the very end product of this degradation. All right, so that's it. That's it for today. So I don't know how helpful was that. So I will continue discussion of this. Okay, I got another question. Degradation of cytosolic protein and extracytosolic proteins are different. cytosolic proteins, yes, there are some cytosolic proteins that helps in the maintaining the structure of the cell. Let's say some cellular cytoskeletal protein. An extracytosolic protein, it can be outside the cell, or it can be some proteins, let's say some of these proteins, which are not meant to be in the cytosol. They're meant to be in some organelle. I'm not sure if the degradation is different between these two exactly. I am not sure what is the difference between the degradation of these two proteins. So what I'm sure that this protein, which is unwanted or misfolded, they need to be degraded, and usually proteasome does the job. Proteasome degrades these proteins into small molecules. All right. Okay. So that's it. That's it for today for this session. Hopefully it was helpful, the discussion of the questions and how you can pick some answers. And we will continue this discussion tomorrow also. And the discussion I have decided to continue like this, like the first, let's say I have discussed the biochemistry and immunology, the very basic, right? The very first few things of the biochemistry and immunology. So every topic I discuss, it will be from the, it will not follow like, it will follow the sequence of your, mostly of your first aid contents, because the first aid you are reading as your main book. So in the first aid, the contents are arranged in sequence. So I'll follow that sequence, that, okay, I'll not jump into the very deep in the beginning. So beginning, it will be very basic of the basics. Then eventually I'll go into the deeper, right? Okay, another question. Proteasome system presents to MHC through TAP. Is it true for this one as well? MHC1, I think MHC1 is associated with TAP, transporter associated with protein processing. That is different. In TAP, the transporter associated with antigen processing or transport TAP, TAP is true for MHC1. For MHC2, the process is completely different. We'll discuss it some other day, okay? It is associated with phagosome, the lysosome, phagolysosome, fusion, then the invariant chain dissolving, and many things. So we will discuss it later. But this one, the TAP, is associated with MHC class 1. And by the way, where is the TAP? You might ask me. Let's just show you. Do you remember this transporter through which this antigen is getting inside the RER, rough endoplasmic reticulum? This transporter is TAP. All right. So TAP is a transporter that is transporting this viral molecule inside the rough endoplasmic reticulum. All right, everyone, whoever is joining through the Facebook and whoever is joining through the webinar, thank you very much for joining in. Please fill out this form, the Google form that will pop up in your window if you're joining through the webinar. You just need to fill up this once, not every day, just once. If you're done filling it once, you are all set. All right, cool. Thank you very much. Have a nice day. See you tomorrow. Goodbye.